Hey, hi everyone. So today we will finish talking about the process of T cell development. Um, and then starting with next lecture, we'll start putting together some of the pieces about how T cells and B cells actually work together to carry out their similar but different enough to be equally important functions in our adaptive immune response. So this is where, as a reminder, the way the textbook is kind of set up, it kind of bounces back and forth, right? We learn about B cells, we learn about T cells, how they develop, and then we go back and start looking at how they actually function together. As a reminder, you do have a quiz this week. And the quiz is on the chapter 6 and 7 material, which is the B cell development and T cell development stuff that we've been talking about these past few lectures. So don't forget, your quiz is due Thursday night, tomorrow night by midnight as usual. Okay? All right. This series of cartoons, I think, is a really useful set of illustrations to talk through. Because what we're building towards is really an understanding about how T cells and other immune cells that present antigen to the T cells really work together, and how that's dependent upon the T cell's ability to properly see things that are presented to it on the surfaces of other cells. Right? We can start off with this illustration on the upper left here. It's a nice micrograph image of a T cell talking to or communicating with some other cell that it's bumped into, right? And it's bumped into it, and there are numerous types of receptor proteins that help actually form a synapse, or a little surface of interaction between the two cells. Importantly, if you squint, you can kind of make out the fact, right, that there's still space between them, because this is not as though the two cells have somehow fused together into one cell. This is just the two cell membranes being able to contact each other so that proteins that are on the surface have a chance to interact. Right? So this presenting cell might be, for example, let's look at this cartoon right here. What if it's a virus-infected cell? Right? We talked about this before, but I think it's important for us to kind of review and remember this. A virus-infected cell, it would have bits and pieces of viral protein kind of floating around in the cytoplasm, right? Simply because you would expect that if this thing's loaded up with virus particles, there's all kinds of molecules from the virus inside of it. And in terms of T cell, the type of molecule that we're talking about that's important are small peptide fragments of the virus proteins, right? So the little red dots here are floating around in the cytoplasm. Remember, we learned that this might come about with the help of things like the proteasome of the cell, kind of chopping up larger pieces of protein to actually get small little pieces. And then those small little pieces, if they're floating around in the cytoplasm, might get transported into the endoplasmic reticulum, where the yellow cell here, the virus-infected cell, right, it's making MHC1 platforms, and those MHC1 platforms can be loaded up with these small red dots inside the ER before the loaded platform is then moved to the surface of the cell. And that's what we see here. Remember, MHC1 in the cartoons, you can tell it's MHC1 because it's made up of this one long yellow part here, this one long yellow chain, and this kind of blob, and this yellow blob that's pressed up against it, right? That's the microglobulin blob. So in this case, if you have a CD8 positive cytotoxic killer T cell that comes by and synapses in the photo up above with this virus infected cell, in this particular case, this T cell receptor on the surface of this T cell is the right shape to detect the red dot being presented to it on MHC. Right? It's up to a different receptor, the CD8 co-receptor, to kind of swing over and actually confirm that this is in fact MHC1. Right? And if the T cell then receives that message, then maybe this cytotoxic T cell will go ahead and instruct the virus-infected cell to undergo apoptosis. Right? In other words, the virus-infected cell should die so that it stops the spread of the virus. Right? On the other hand, what if the presenting cell is what we call a professional antigen presenting cell, like another immune cell, such as a B cell? A B cell that has on its surface, right, IgM that this B cell made itself, right? And when it was released from the bone marrow, it had this IgM on its surface. It passed the tolerance training to make sure that this IgM wouldn't bind to any normal signals in the body. But now this IgM is binding to sort of this protein from, let's say, the virus, right? And so as a result, because the B cell is constantly pulling in these IgMs, just like any other protein on its surface, constantly recycling them, it will pull in also this red hibiscus-shaped protein from the virus. And when it gets processed, the protein gets processed in the B cell, right, it will be loaded onto MHC2, right? Because in this case, the loading of the MHC takes place in vesicles in the cytoplasm, right? Not inside the endoplasmic reticulum. So the red dot gets loaded onto MHC2, and then maybe a CD4 positive helper T cell comes by, synapses, its T cell receptor recognizes the red dot in the context of being presented on MHC, and then the CD4 co-receptor that helper T cells have comes over and confirms that this is indeed MHC type 2 platform that's presenting the red dot. And as a result, this helper T cell releases these green triangles, which are different cytokine messages that encourage this B cell to fully activate, right? And this B cell might divide more, and then some of them may even become plasma cells to go ahead and release this IgM, right, in the form of antibodies. So the thing that I want you to keep in mind is in both of these cases, it might be the same virus that we're talking about, right? It's just some of your cells got infected by the virus. Others of your cells can detect the virus while not being themselves infected, right? And so the red dot in both of these cartoons could be actually the exact same red dot, right? But depending upon what type of T cell ends up recognizing the red dot and being able to properly confirm whether the red dot is being shown to it on MHC1 or whether the red dot's being shown to it on MHC2 will make all the difference in the world as to what happens next. The illustration in the upper right gives you the true three-dimensional structure of these interactions. So, I mean, it's one thing to see these cartoons drawing them this way. This is what we've gotten used to seeing. But hopefully by now, all of you like seeing the real three-dimensional shapes, right, to make sense of what we're seeing in the cartoons. Right? So here we have two different interactions. So even though this is drawn as though it's taking place between, just two cells here, right, the truth is this lower panel, right, is showing you that there's a cell that's showing something like the red dot. Here's the red dot, the little peptide, right, on MHC class 1. And then we'll imagine there's another cell that's showing it on MHC class 2. All right. But let's look at this. So this sort of peach pink part is the MHC. So this is MHC class 1. So down below, there's a reminder that the cartoons in the book draw it this way. One long yellow chain that kind of wraps from the upper right down to the lower left, and this beta microglobulin protein crammed up against it, right? So this long chain would be sort of the peach-colored part that you see here, right? And then the microglobulin blob would be this pink thing pressed up against it. Okay. Here's the T cell receptor on a T cell. So this upper gray membrane is the T cell that's synapsing with this cell, right? So this space in between the two gray here is this space that you can barely see between the two cells here. But you can see here's the T cell receptor. It's an alpha beta T cell receptor. So you have a alpha chain, beta chain. I don't know which is which. It's just colored light blue, purple. But notice this T cell receptor is the right shape to bind to the red dot, right? Or the peptide here. But also, this helps you appreciate how much of that interaction is actually nothing to do with the red dot. It has to do with the TCR recognizing that this is MHC, right? So that's why the CD8 co-receptor is so important, right? Because this interaction between the TCR, the blue and the purple, with the peach colored part of the MHC, right? It can't tell if this is MHC1 or MHC2 that it's interacting with. It's the CD8 co-receptor represented by these two green blobs here, right? That recognize and confirm that this is the right shape to be MHC class 1. Right? So that's why you get then the combination of the TCR signaling plus the CD8 signaling that then allows the cytotoxic T cell to activate. On the other hand, here is a case where the red dot or the peptide is being shown on MHC class 2. You can see the structure of MHC2 is different, right? Because it's made of just simply two chains here, the yellow and the orange. Okay, so that's kind of this up here. But again, here's a T cell receptor. Again, it interacts, recognizes the red dot in the context of it being MHC. And here's the CD4 co-receptor, all right, the CD4 co-receptor that swings over and confirms that this is in fact the right shape to be MHC2. So that's what's happening on the lower left of the slide, right? So you can see why that it's important for both the T cell receptor to recognize and like what it sees, but also the co-receptor protein, right? And the CD8 cells only have CD8 co-receptor on their surface. The CD4 cells only have CD4 co-receptor on their surface. And that's how these T cells can either help a target cell or kill a target cell. All right, so let's look a little bit more at this whole idea of how a developing T cell gets to that point when it really decides to be either killer or helper, right? Because up until now, when we've talked about the development of T cells, the pathway has been the same for both. So we studied this last time. We said that, remember, this green precursor cell was released by your bone marrow. It's an uncommitted progenitor cell. It doesn't know what it wants to be, right? Maybe it wants to be a natural killer cell, whatever, but it so happens it made its way into the thymus. So the thymic epithelial cells that began interacting with it encouraged it to consider becoming a thymocyte on its way to becoming a T cell. Right? And one of the key signaling events is that Notch1 signaling that we looked at last time. That's the peer pressure, right, that gets this uncommitted progenitor cell to really want to become a T cell. We learned last time that the VDJ process begins, and in this thymocyte, the VDJ, the oven mitts, right, begin to go to work, the RAG protein complexes. They'll first try to rearrange the beta chain, the gamma chain, and the delta chain. And if the gamma and delta chains actually work and they make gamma and delta chain proteins, then the T cell just becomes a gamma delta T cell, and its time in the thymus is done, right? It can now be released to go do its gamma delta T cell functions, which have to do more with tissue maintenance rather than looking for novel antigen signals. However, we said if the RAG oven mitts succeed in making a beta chain that works, then the thymocyte is on its way to becoming an alpha beta T cell. Right? So at that point, the thymocyte then starts making both CD8 and CD4 because it has no idea what it's going to become. Right? But it wants to have those co-receptors ready, because those co-receptors will help it make its ultimate decision which way it wants to go in the end. Importantly, the RAG oven mitt starts up again, right, and starts making the alpha chain to go along with the beta. But at the same time, it has access to the gamma and delta loci, so it could very well just this time make those chains. And if it does make a gamma and delta chain this time, right, this is the second opportunity to make a gamma delta T cell. And again, that gamma delta T cell will then just go off, right? But if it makes the alpha chain, now we're set. We have the alpha beta, the yellow green T cell receptor on a double positive, CD8 positive, CD4 positive T cell. So now you have to start making the decision. Which type of T cell is this going to be? this figure here is kind of a nice way of thinking about this process leading up to this point, right? So this type of figure, what they're talking about here is the different sort of status of the developing thymocyte from being double negative, meaning it doesn't have CD4 or CD8, to being double positive towards the end, where now it's making both CD4 and CD8 to get ready to make a decision about what it's going to be. And then down here, the table reminds you about what's going on inside as far as VDJ recombination and the presence of different proteins, right? So as the double negative thymocyte begins to rearrange its beta, gamma, and delta chains, what you see are the RAG proteins, the various proteins that make up the RAG oven mitt complexes, right? This red down here means you see that those proteins are being made in the cell, and then the RAG oven mitts are being made, and the oven mitts are going to work doing the rearrangement. Right? So it starts off by making the DJ rearrangements, choosing a D block and J block of the beta chain, right? So that's why you begin to see, right, the rearrangement of the DJ blocks here. And then once that's done, the RAG oven mitt sort of pauses. So the production of the RAG oven mitt kind of goes down because the cell wants to just make sure at this point, have an opportunity to make sure that the beta chain looks good, right? So remember, it does that by doing a little quality control check. It has this little gray PT alpha protein that binds up with the beta chain of the pre-TCR, right, that's forming, and make sure that it's okay, right? So this PT alpha here checks it out. If that looks good, right, then you proceed with moving on to making the alpha chain, right? So that means you can see here what's happening. The RAG oven mitt proteins get turned on again, right? So now you need the oven mitts again. So the cell starts producing these, and you do the rearrangement, the VJ rearrangement of the alpha chain, right, along with the gamma and the delta. Right? And then because that's where the cell is headed, right, it's headed towards making an alpha beta receptor, suddenly you see on the surface CD4 and CD8 co-receptor proteins, which up until now, right, were not present. At this stage, the cell starts making them. Right? So ultimately, though, the final decision that has to be made, right, is here. Selecting whether the cell is going to stick with being CD4 positive, right, and give up the CD8, or whether it's going to stick with being CD8 positive and give up the CD4, right? So the end result should be a T cell that is only single positive for CD4 or CD8, thereby making it either a cytotoxic or a helper T cell. So let's think about this. This process then is more complicated than how our tissues make B cells, right? How our bone marrow makes T cells, because there's a lot more subtlety here in the way you're selecting for things. The development of a T cell requires different steps along the way where you are positively selecting for the developing thymocytes that have certain features that you want them to have, right? While at the same time negatively selecting or rejecting certain T cells that have features that you don't want them to have. And what do I mean by features? I mean, it all has to do with how these T cell receptors are going to work to interact successfully with things presented to them, right, by MHCs. So let's use this kind of analogy here of some German Shepherd puppies, right? So at the start, when the precursor of a thymocyte arrives into the thymus, right, and it's uncommitted, it has no idea what it's doing. It was just wandering around, and now it's in the thymus, right? It's kind of like these little few-week-old puppies here, right? During this time, if you're going to train these puppies, train them to be dogs that, hopefully provide a certain job or task that they're trained to do, maybe this selection starts very early by maybe doing some negative selection, right? So you need to make sure that certain puppies that behave well are the ones that look like they're good candidates for further training. So at that point, you can say, well, I'm positively selecting for certain puppies that have good behaviors that I like, while at the same time negatively selecting for puppies that, are a little too rambunctious or whatever, right? Then as they get older, right, the training gets a little bit more sort of detailed, right? Because what you want to do now is very carefully make sure that these dogs are actually properly trained not to overreact against certain situations, right? So to be honest, I don't know what's going on in this picture. This little cat is like strutting in front of these dogs. I don't know what happens like a moment later, right? Maybe all mayhem breaks loose. But the point is, up until this point, all of the T cells have gone through the same sort of combination of positive and negative training to lead to this point. And they are at this point double positive, right? They have CD4 and CD8 under surface. So now the critical decision has to be made, right? Which type of T cell is it really going to be, right? And that's ultimately the decision that's going to have dramatic impact on what this T cell ends up doing as its job, right? Is it going to be a cytotoxic T cell that kills other cells, or is it going to be a CD4 positive helper T cell, right, that encourages and supports the activity of other cells? So let's think about how this selection process takes place. So it's all going on in the thymus, right? So that's why I'm showing you this again. And as a reminder, in the thymus, you have these different compartments. And in one of these compartments, in the cortex region, the sort of bluish region, you would have those brand new precursor cells arriving, receiving kind of some peer pressure by these orange, epithelial cells that are signaling them to become thymocytes, so that these then start doing their VDJ recombination and they make their T cell receptors, right? So then they migrate down to this medulla region of the thymus, where then different cells, like these darker orange epithelial cells, right, begin to do the task of helping them finish their training and decide if they're going to be cytotoxic killer T cells or helper T cells. So let's talk about this positive and negative selection, right? One of the reasons why this process is so much more involved in T cells compared to B cells is, remember, a B cell just has to make an immunoglobulin on its surface, right? An immunoglobulin that looks like an immunoglobulin. And just as long as that shape of that Ig doesn't cause problems by binding self antigens, then whatever, right? The B cell is let loose into the blood to undergo additional screening there, and then the B cell can go into something like a lymph node and be rewarded and activated and turned on, or I shouldn't say turned on, but matured, right? So that its IGs on its surface then are ready to work, and then the B cell goes into circulation. However, remember, T cell receptors are limited in their ability of what they can bind, right? Not only can they only bind to small peptide fragments, the little red dots, but those small peptide fragments have to actually be presented to them by MHC platforms. And as I already told you, right, so much of the contact between the T cell receptor and an MHC platform presenting a red dot, right, is actually interactions between the TCR and the platform itself. So that means, as part of the training of a thymocyte, when it makes a new novel T cell receptor shape, that shape still needs to be the right shape to actually properly bind to MHC. Because if that T cell receptor can't even bind to MHC, then what good is it going to be, right? So one of the first steps after a thymocyte has successfully made, let's say, an alpha beta T cell receptor here, is it needs to be selected for whether or not this T cell receptor can bind to MHC. So you can see in this scenario here, right, this developing thymocyte, its T cell receptor is the wrong shape to bind MHC. So this epithelial cell is one of these cells inside the thymus that's now presenting. So this would be in the lower medullary region of the thymus. Right? The T cell over here successfully made its TCR in the cortex region, and now it's getting sort of trained at the bottom. In this case, these epithelial cells will present MHC, right? And these cells will present both MHC1 and MHC2 on their surface, just to see whether these newly developed thymocytes with T cell receptors can in fact properly bind to MHC. Right? This particular thymocyte, it's too bad. Its shape that it came up with its T cell receptor does not interact with MHC. So let's face it, right? This T cell is never going to be any use if it can't interact with MHC in order to see things that cells are presenting to it. So as a result, if there's weak or no binding to the epithelial cell, this thymocyte does not receive any encouraging cytokine signals to keep it surviving. And as a result, it just dies. Right? On the other hand, this thymocyte down here, its random TCR shape that it came up with, is pretty good at binding MHC. Right? So you have moderate or potentially stronger binding. That means what? This thymocyte will spend more time pressed up against or close to this epithelial cell that's sending out clouds of encouraging cytokines, right? So this thymocyte will receive survival messages that keep it going through this process, right? So if we think about the whole German Shepherd cat analogy, right? What we don't want is we don't want the German Shepherds to completely ignore the cat, right? Because as part of what these German Shepherds, you're training them to do, is they have to work with cats, right? That doesn't make any sense. I don't know why you would train German Shepherds to work with cats, but just go with it, right? What you want is you want a friendly interaction, right? Because only these German Shepherds are the ones that can do the job that you have in mind for them. Right? All right. Importantly, since the TCR is just all random shapes, there's no telling whether the TCR of this brand new thymocyte would preferentially interact with MHC1 or MHC2, right? But whichever one it interacts with first and has that decent interaction with, that will dictate from now on that developing thymocyte, right, is heading towards being the type of T cell that interacts with either MHC1 or MHC2. In other words, the TCR, the receptor, will be restricted to just binding to that class of MHC. Let's look at some of these structures a little bit more closely here, right, to really appreciate what I'm talking about when I say it's so important that the TCR can actually recognize the MHC shape. Let's start with this cartoon in the middle over here. Right? So here you have an alpha beta TCR on a newly developed T cell or a thymocyte. So it has the alpha chain and has the beta chain. And remember, each part has a variable region and a constant region, sort of like the immunoglobulins that we learned about before, right? On the bottom is the MHC class 1 that this TCR happens to have bound to, right? And the MHC class 1 is presenting this yellow dot, which is the peptide of interest, right? That this presenting cell has decided to load up on the platform for a T cell to check out. So even in this illustration, which we've seen a lot of, you can already tell that much of the surface that's interacting between the TCR and the MHC is just between the TCR and the MHC itself, right? Only part of it, like this curved part in the middle, actually is the important part for being the right shape to bind this yellow dot. Let's move to the left. This is the actual three-dimensional structure, ribbon diagram of the polypeptide backbone of these proteins, right? So this is kind of cool, right? Because you can relate this to the cartoon in the middle here. So on the left, again, is the alpha chain, right, with its two parts, the constant and the variable part. On the right is the beta chain, the constant part and the variable part, right, just like you see in the cartoon here. But what's neat about this is, remember, the variable antigen binding ability that comes from VDJ recombination really has to do with certain specific loops of the proteins that define these hypervariable regions, just like in the immunoglobulins of B cells. Look at this, right? The three-dimensional shape that's determined by this kind of random configuration of loops here determines the three-dimensional shape that this T cell receptor can bind to and recognize. Right? In this case, the lower part of this figure is the MHC class 1. So on the cartoon here, remember it's one long chain that starts from here and goes down to here. That's the alpha part. And then you have this blob, this globular microglobulin, this beta microglobulin here. So that's what we see here, right? Starting from here is the long chain that wraps around and goes down. And then here is that beta 2 microglobulin blob that fits in it, right? So look at this interaction surface. Among the different loops that are part of the T cell receptor that are colored like two shades of purple. There's a dark purple, a light purple. It's kind of hard to tell. And then two shades of blue. There's a light blue and another dark blue one in the back there, right? And these yellow ones that represent these loops over here, it's really only these yellow ones that determine whether or not the TCR can recognize the shape of the yellow dot. So in this case, the yellow dot is shown as this yellow short peptide, right? Because that's what it is. It's a peptide. It's not really a dot, right? It's a short peptide. So look, though, at how much of this interaction is actually the purple loops and the blue loops playing a role in detecting this. To see this easier, what if, instead of looking at this from the side, what if we were to imagine that we were looking at it from the top, looking straight down the middle at the interaction surface between these two molecules, right? That's what you would see here, okay? So what we're looking at are the solid sort of shaded spheres in the background are the MHC1 part. So it would be the upper surface of this MHC1 over here with the yellow squiggly peptide in it. So that's what you see. The sort of reddish or maroon background there and the bluish one here, those are the two sides of the cleft of the MHC that are holding in it. You can see the yellow, you see the sort of, yeah, I mean, it's in the background, but this yellow kind of squiggly thing going through there is the peptide that it's holding, right? So again, you can look up here as a reminder, right? MHC1, it has the two sides of the cleft. MHC2, right? It also has two sides of the cleft. And in here would be the peptide laid across, which isn't shown in this set of cartoons on the right. The outlines on this are the footprint, right, or the shadow of where the T cell receptor is coming down and pressing up against the surface. And again, the purple shadow and the darker purple outline and the lighter purple outline, darker blue outline, lighter blue outline represent where these loops over here are coming into contact, right? And you realize that the yellow ones in the middle are the ones that actually determine whether or not it fits the shape of the peptide. So much of this interaction is just the T cell receptor properly recognizing MHC. Right? So that's why you're selecting for T cells that have T cell receptor that can do that. I want to back up just a moment because I wanted to point out an important fact that I missed. I think it should be kind of obvious that if the shape of the TCR, like those loops at the end, came about through the sort of random VDJ recombination process, you say to yourself, well, what are the chances then that they actually are the right shape to interact with MHC at all, right? The chances are really slim. That's why, right, after a few days of this type of selection process, only about 2% of developing thymocytes that successfully made a T cell receptor actually get positively selected for, right? That means the other 98% of those thymocytes that went through that whole process of being encouraged to become a thymocyte and doing VDJ recombination to make a T cell receptor, right? They're garbage because their T cell receptors don't bind to MHC. And if they don't bind to MHC, they're never going to be able to work as a T cell, right? Another example of how crazy inefficient our adaptive immune system is. But why did we evolve to do it this way? Because we have no other choice, right? Okay, so the next step, though, this is where it gets even more kind of like a gray area, right? We said it's important that a brand new T cell receptor interacts with MHC, But what about if by chance the shape of that T cell receptor not only interacts with MHC, but interacts with MHC a little too perfectly, right? Almost to the point where it doesn't matter what peptide the MHC is showing, this T cell receptor will happily bind to it. That is clearly a problem also, right? Because such a T cell would be very dangerous. It would interact with any cell just because that cell has MHC on its surface, and then that T cell would activate, right? And it would cause all kinds of weird autoimmune reactions as that T cell then tries to get the rest of your immune system to just attack any cell that has MHC on it, right? What a catastrophic failure that would be. So that means that the T cell receptors have to be selected for that bind moderately well, but not too tightly. So here, let's revisit this. This was our happy little binding between the German Shepherd and the kitten here. So you have moderate binding, but not too tight, right? And therefore, that cell is allowed to live, all right, and continue on, because the hope is that at some point in the future, if it does go to work, right, that gentle moderate binding with the MHC will be greatly enhanced if that MHC is showing a peptide that really tightens the fit of that TCR with it, right? Because that TCR happens to have the right shape to also recognize that peptide. However, if you have thymocytes that bind too tight, almost too aggressively, right, you don't want that either, right? Because you don't need to have this going on, right? So those super tight binding ones, they die, right? And I know you're thinking, that's so weird, because how do these cells know the difference between moderate binding and tight binding? Again, it just has to do with how much time the thymocyte is exposed to messages coming from the dendritic cell that it's interacting with, right? If it spends enough time to kind of receive the survival messages, but it's not binding so tight so that it actually floats away also, then it's okay, right? But if it binds too tight, then it's like it overdoses on those types of cytokine messages, and that actually causes the negative effect, right? It like ODs on those messages from the dendritic cell, and then it dies, right? So I think that's amazing when you think how subtle all of this is. And yet, you know, even though this sounds really subtle and sort of wishy-washy, you know, this is how we evolved, and this is critical for your ability to actually be sitting there right now listening to this, right? Because if any of this failed in your body 20 something years ago, right, you wouldn't be here. Okay. All right. So to summarize, we have an example here, two different double positive thymocytes, right, that have successfully made TCR, right? So their surface is covered with TCR. So here's one TCR, here's another TCR over here, right? And at this point in the development, they don't know what they're supposed to become. So they have both CD8 and CD4 on their surface, right? So the CD8 is represented by these molecules up here, the sort of lollipop looking ones. And then the CD4 is represented by these more, I don't know, thicker looking single strands, right? Single molecules. So depending upon which type of MHC the TCR has a chance to interact with first and interacts with favorably, right, that will then determine if the cell becomes CD8 positive or CD4 positive only. Right? So in this case, what it means is this double positive thymocyte on the left, right, it binds with its TCR to the red dot on MHC1. The CD8 receptors on its surface come over and confirm, oh yeah, we've bound to MHC1. So what happens? This then sends a signal into the nucleus of the cell that says, stop making the other co-receptor, stop making the CD4, and just stick with making CD8 from now on. All right. On the other hand, this other thymocyte over here, its T cell receptor binds to MHC class 2. So its CD4 co-receptor confirms, yes, we preferentially bound to MHC class 2. So therefore, this cell then turns off any further expression of CD8, and it becomes CD4 single positive. right? And what I think is remarkable about this, right, is this blue thymocyte up until now could have been exactly the same, right? And it just happens to be which MHC its TCR preferentially interacted with. And yet, the difference in function between an aggressive cytotoxic killer T cell versus a helpful and supportive CD4 cell, right, couldn't be more different, right? So what does that mean? How could it be that one's a killer and one's a helper? It all has to do with, from now on, what types of cytokine proteins the two different T cells make. Whether it makes cytotoxic cytokine proteins that, if they're sent out and received by another cell, tell that cell to die, right? Or if the types of cytokines it makes is instead encouraging cytokines, right, that encourage a B cell to become a plasma cell, or encourage a macrophage to increase its phagocytic activity. Right? As we will learn, the cytotoxic T cells are pretty straightforward. I mean, if they pass all their training, they go off and just start killing things if they encounter them, right? So they're kind of single-minded, right? Whereas the helper T cells, anytime you're thinking about helping something, that's a more complicated kind of touchy-feely kind of situation, right? So not surprisingly, a CD4 helper T cell will further differentiate and become different subtypes of helper CD4 cell, each type specializing in helping or supporting a different type of immune cell. Right? The first example of this is, I want to show you this orange CD4 helper cell here, which we call a regulatory CD4 cell, or a Treg. Right? The job of the Treg is to actually keep other T cells from getting too hyper and overreacting. Right? What do I mean by that? If you think about it, your T cells that are in your system right now, in your lymph nodes, in your blood, going back and forth, right? They have the potential to bind to all kinds of things, in varying degrees, right? So even if they bound to a certain antigen target, right, maybe it's just they would bind certain ones stronger than other ones. Right? As a result, at any given time, even if you're not really facing an infection, there's always going to be some level of what we can call noise, right? Of T cells just kind of like improperly activating, right? Because something has bound to it. And if you always were ready to overreact to that noise, then your immune system would constantly kind of be turned on, right? And that's not good. So the way our immune system evolved is to accept a certain level of noise, of sloppiness of our T cells, right? And only try to respond to things, signals that rise above the level of that noise. In other words, if T cells really start reacting against something, right, in a way that takes them above the normal humdrum noise level of the activity, right? So that's the job of the Tregs. So the Tregs are helper T cells that will bind to something like an antigen presenting cell, as shown down here, right? And they bind to MHC class 2, and they have CD4 on their surface because they are technically CD4 positive helper T cells, right? But when they bind to an APC, they send out clouds of cytokines that actually inhibit other T cells from activating. So this other blue CD4 T cell comes and also binds the same thing here, right? So this T cell wants to activate, but because it's being suppressed by these Tregs, right, this particular T cell might not activate that much. But if you get at a certain point where you have more and more of these blue T cells coming and binding, right, because you have more and more antigen presenting cells showing this signal, then that level of activity rises above the noise, right? And your immune system will react to it. So this idea of noise is kind of interesting. I think about it like your car radio. When you're out driving and you have your radio tuned into a station and the music sounds really good and clear, and then suddenly you go into a tunnel, right? most of the time, you start hearing like the music start fading, but at the same time, you hear a lot of noise, a lot of hiss or whatever coming in until eventually the signal just breaks up, right? What is that hiss? That hiss is the background signal, right? Your antenna of your car is always receiving all kinds of noise, right, through different radio transmissions, and just the electronics of the radio itself, right, introduces a lot of hiss and noise. But your car radio, right, has the ability to essentially filter out that noise, and any stronger signals that it's receiving, like the actual music from, from the radio station, right, it preferentially amplifies that. But when you go into the tunnel, right, you lose the ability to receive the good signals, and your radio then tries to amplify what it can work with, and so all the noise level becomes more audible, right? So think about it that way. Your immune system is always trying to sort of ignore or suppress the noise of your T cells being sloppy. But if strong signals happen, right, like enough of these T cells really begin to signal, then your immune system reacts to that, right? And so our Tregs are very important for maintaining that noise filtering to give us nice, clean music, right? Last slide for today. So T cells also have to undergo self-tolerance training, just like B cells, right? Or I should specifically say alpha beta T cells, right, have to go through self-tolerance training. The gamma delta T cells that took a detour off of the T cell development pathway, they don't have to do this, right? So this training takes place in the thymus before the T cells are allowed to leave, right? And it involves a remarkable mechanism, right? Remember, the whole point is, because the T cell receptor was made randomly from VDJ recombination, and you're cooking up, millions upon millions of different shaped T cell receptors, there's a very good chance that the T cell receptors will bind to something that's being presented by a totally normal, healthy cell in your body on its MHC, right? So the T cells are not allowed to leave the thymus until they've been screened to make sure that this won't happen. Why? Because unlike the B cells that are released from the bone marrow and have a chance to continue their training in the blood because their immunoglobulins aren't properly wired up yet until they get to something, a lymph node, the T cell receptors on a T cell, they're ready to go, right? So the moment a T cell is released from the thymus, there is the potential for it to start interacting with other cells it's bumping into in the blood, right? And if those cells are presenting, let's say, totally normal peptide, and this T cell receptor unfortunately recognizes that, right, that's bad. So that would be another big autoimmune failure, right? But how do you actually make sure that these T cells, right, are so ready to be released from the thymus? The thymus epithelial cells have a particular protein factor that's expressed in them, known as AIR, or autoimmune regulatory factor. This protein in the epithelial cells of the thymus is a transcription factor, right? So in other words, it goes around then in the genomes of the epithelial cells and randomly starts turning on expression of any genes, all right? So that proteins that might normally have no business being part of a thymus, like a protein that's expressed by heart muscle or bone or by other types of tissue, right? At least in these epithelial cells, because of this AIR transcription factor, you have little bits of these proteins that are made at random. Right? And then these epithelial cells are busy showing these little bits on their MHC that they're using to train the developing T cells. Right? So therefore, just in the thymus alone, a new T cell is able to experience many different potential body antigens without ever leaving the thymus. Right? And so this is considered the central tolerance training of the T cells. Right? And so I think it's kind of neat when you think about that evolutionarily, right, that's how these developing T cells get a sneak peek of a multitude of different possible normal signals that they will encounter later when they go to work in the body. And you make sure that they don't react to them when they're still in the thymus, because at least if they're still in the thymus, they can be either encouraged to do a little bit more recombination to try to get their T cell receptor not to be so sensitive, right? Or the T cells can just be eliminated and not allowed to go to work. Okay. All right, let's go ahead and stop.